So, welcome back to our quest to understand the molecular processes of genetics. We have been seeing various processes and we saw transcription in our earlier discussions. The next gradual step which the cells they carry out is translation. So this is going to be a bit more interesting and a longer type of discussion. So I have broken this lecture into two parts. Part 1, I would like to detail about the tRNA molecules or the Crick's adapter molecules, and part 2 we will be dealing with the translation process as such. Translation process needs to know the significance of tRNA molecules. As students, you should know the importance of tRNA molecules. Even though if it is not there in the syllabus, as I said, syllabus is insignificant here. The process is important and the requirements which are there gradually one by one we are studying in detail. So before that, proteins, we are trying to understand the protein synthesis. What are proteins? Proteins are the workhorse molecules found in the organisms. These are the main molecules which are responsible for metabolism, and metabolism governs life. So protein synthesis is life, I can say. Everything depends upon protein synthesis because metabolism depends upon enzymes. Each and every step of a metabolic reaction is catalyzed by enzymes, and all enzymes are proteins. So there will be as many as genes, as many as proteins are present, and all these are the sequences present on DNA which are copied down as the sequences on mRNA and then read in the form of proteins. So you can say these are the blueprints for life, and the blueprint for proteins is coded in the DNA. So therefore we call DNA as the blueprint of life. There are many uses of proteins. The bacteria, they use proteins for many purposes. Formation of a structure like flagella, enzymes, for transport of molecules, transporting themselves from one location to another location by flagella, and responding to various environments like chemotactic, phototactic, aerotactic. So every action requires one way or the other proteins. In eukaryotic cells, you can see in humans carrying gases, hemoglobin, a protein which is iron containing protein which is responsible for carrying oxygen. So transport of gases, enzymes, then structures, the spider web, you know, that is a protein, and formation of structures, antibodies, defense mechanism, then storage of protein, movement of protein. All these life governing processes are mediated by proteins, and that is the significance of protein synthesis. The protein synthesis main stage, it begins, if you just go to see the beginning of protein synthesis, you go back and back and back, you will go and end up in evolution. So somewhere we have to start. So we start with transcription, formation of mRNA, and now the main what you can say peak of protein synthesis, the peak point of protein synthesis, or the main production stage and the factory where it is made is the ribosome, and that is the process of translation. We have seen the structures of ribosome. So this ribosomal structure which is there, this encompasses two subunits. We have seen the 50s and the 30s. 50s is the main hall which accommodates 3 tRNAs. My point of discussion here is today tRNAs, that is the transferal RNA. One question may arise in your brain: if all these are RNAs, how come that this mRNA is a dynamic structure and these two are stable structures? That is understood very well. In the subsequent slides you will understand that the nitrogen bases present in rRNA and tRNAs are modified in evolution. Okay, they are not that normal or routinely bought from the nucleotide pool and structure is formed like mRNA. No, they are stable structures. They contain some modified bases, and these bases have been modified during evolution. Of course, they are formed from certain gene. I will come to that, but let us try to understand the structures of tRNA. these tRNA which accommodates in the 50th subunit, and each tRNA brings an amino acid, and this process is called as translation. So I'll come to that. Let's just move ahead. All this takes place by reading the nitrogen base sequences on the mRNA, reading of the triplet codon. We have seen how these codons are arranged on the mRNA, copied down from the DNA and read in triplets. We saw this in the characteristic features of the genetic code. There are three potential reading frames in any mRNA molecule. See this, the first one. Let's call this as frame 1. This is the same mRNA sequence, that is GCU, UGU, UUA, CGA, AUU, AA, for your understanding. Hypothetical mRNA chain. Frame 1 is also same, frame 2 is also same, frame 3 is also same. Now all it depends upon which is the first nitrogen base which rRNA recognizes, so that the subsequent nucleotides will be read in triplets. So see here, the frame 1 GCU is recognized. So as it is a non-overlapping and a comma-less code, subsequent nitrogen bases will be read in triplets. So this is a first polypeptide chain formed. GCU will code for alanine, UGU will code for cysteine. But the whole scenario changes if instead of G here, C is read as a first base. See here now in the second reading frame example, this is a second example of a reading frame. All are possible, but in all the three reading frames, one thing is common, that it is a comma-less code and it is a non-overlapping code. So CUU is the second possibility. So instead of alanine, leucine will be there, valine will be there, tyrosine will be there, because the whole reading frame has shifted to one base. This also is a possibility. All these are done by a combination of ribosome, and the reading here is done, which I am showing here, is done by tRNAs. So you have to understand what is tRNA, its structure and function. See the third reading frame. Why three reading frames? Because triplet code, there is a possibility with that this will be the first base to be read, or this will be the second or third. So third, let us see here, GCU, third will be UUG here, UUG. So if UUG is read as the first codon, leucine, phenylalanine, serine, tyrosine, a third type of polypeptide is changed. So from one mRNA, you see three different types of polypeptide chain can be synthesized. Possibility is there, and this possibility has to be in combination by the reading capacity of the ribosomes and the adding of amino acid capacity by the tRNAs. However, remember, out of all these three possibilities, only one reading frame is used for translation at a time. Only one reading frame is selected, a frame which contains AUG. So you see, there may be ambiguity here. Which will be the polypeptide chain? It may start, but all these are called as nascent polypeptide chain or immature, premature polypeptide chain. The real protein synthesis begins when the ribosome encounters the first AUG codon. So ribosome may start, once it binds to the mRNA, it may start translocation. Translocation is the movement of the ribosomes. It may start the translocation. It may form the proteins, but that amino acid chain is of no use till the first AUG codon is reached. From there, the actual protein synthesis starts. What happens with this small extra tail of amino acids? It is processed, removed later on. That is called as the post-translational processing. So triplet codons are read in a non-overlapping mode, comma-less manner. Rarely mRNAs are read in more than one frame. As I said, these are the three possibilities, but not all, only one is selected. And like this, the frame shifting example which I have given is very uncommon. It is rare, it doesn't take place. So all this is felicitated by the help of a ribosome. As you can see, this blue structure is the 30th subunit and yellow structure is the, blue structure is the 50th subunit and yellow structure is the 30th subunit. It contains sites for 3 tRNAs. This green and pink molecules are tRNAs, and on the tip it has amino acid chain. At the link at the bottom, I am giving you some links of this video. You can visit there and see some beautiful animations. This whole process of translation is well understood if you see the animations. But first listen to my lecture and then go for the animations. So this is how the ribosome accommodates the tRNAs for the synthesis of polypeptide chain. So what is this tRNA? How were they discovered? First of all, tRNAs are RNAs, okay, but these are stable structures like the rRNA. What makes it stable? The internal folding, the secondary structure. Secondary structures formed due to internal complementary bases. Means you see here, if this is the tRNA, it starts, let's read from somewhere, 3 dash end, A, C, C, A, C. If you label this 1, 2, 3, 4, 5, 6, 7, 8, 9, so on and so forth, you go on. Let us say if there are 100, this will be 95, 96, 97, 98, 99 and 100. So 96th and 10th nitrogen base, they can form a bond. UU will not form, so there is a small gap. GC will form, CG. There are internal complementary bases, and where there is no complementation, it forms a loop-like structure. It forms a loop-like structure, and where there are complementation, it forms the stem. So there are 3 stem and loop-like structures. I will come to the actual structuring, but how was this discovered? Initially, during the discussion where it was, there were 2 schools of thoughts. That one believed that DNA is the genetic material, other believed that proteins are the genetic material. During that time, it was not known that there is an intermediary molecule or molecules like mRNA, tRNA and rRNA needed for protein synthesis. All the scientists, they believed that DNA itself oozed out proteins. The whole DNA molecule, proteins oozed out of it. They thought like that. That was very natural. Why? Because eukaryotic system, if you see, they contain histones. Histones are DNA wrapped around proteins. So if I just break out a eukaryotic cell and see the DNA, I will get lot of proteins along with DNA. So my thinking will be but natural that this DNA is releasing proteins. Today we know that is not the case. In eukaryotes, histone proteins are needed to maintain the structural integrity of the DNA. But that was the hypothesis. But Crick, Francis Crick, in mid 1950s, he together with the scientists who believed in the central dogma of molecular biology hypothesis, he formulated that. He said that DNA itself cannot directly form proteins. There is some intermediary molecule, he called it as the adapter molecule. He said there are some adapter molecules which mediate the protein synthesis. Crick, in his adapter hypothesis, he proposed that there are small RNA molecules. He did not know they were tRNA, rRNA and mRNA, but he said there are some small RNA molecules which act like adapters, and that receive some specific amino acids attached to these adapters. They interact with some another type of RNA called as messenger RNA today via triplet codon. This was his adapter molecule hypothesis. See the vision which they had. Continuously, when you study something in great detail, continuously when you focus on one particular hypothesis or one particular subject for long, you become a master in that, you become a doctor in that. To become a master, bachelor's degree is just the beginning. To become a master in subject, you will have to focus in one single subject. And Watson and Crick did that. When they did that, these adapter molecules, later on the scientists, they studied, and these adapter molecules came to light, and they were seen to participate in the protein synthesis. Subsequently, these adapter molecules were identified today, which we know as tRNA molecules. In 1950, Crick put up this hypothesis. 2 years after that, in 1952 to 58, after 2 years, the thinking and work began on these lines. Earlier they believed that DNA is the center stage and directly proteins were formed. But later on, after this hypothesis, in 1958, after some 6-8 years of research, Hogland and Zemechnik, 2 scientists, they discovered the tRNA molecule. Their main work was with amino acid activating enzymes. amino acid activating enzymes which activated the amino acids, so that these activated amino acids which combine with certain molecules, the tRNA molecules, which eventually form the new molecules. So today we know that they were working on amino acid tRNA synthetase, and this charges the tRNA. tRNA, there is amino acid bound to it, and this brings at the site of protein synthesis. So this is the structure of tRNA molecule. The structure which you just saw here, this is called as the clover leaf model. It looks like a clover leaf, botanical leaf. So what makes it a permanent structure? Most of my students, they have been asking, sir, why these two are stable structures and why mRNA is an unstable structure? And they answer for themselves. They come to know for themselves very correctly that mRNA is required again and again. So these are required again and again. But the mRNA which is required again and again is required in a new combination of nucleotides. These are not needed. They are needed in the same type because they contain an anticodon site. This anticodon is 3 nitrogen bases which is complementary to the 3 bases on the mRNA. So this is fixed, and there is an amino acid binding site at the 3' hydroxyl end. And exactly opposite to this cloverleaf structure formation, you can see here in figure A the simple form of this. There is a stem and loop. 3 stem and loops are there. One stem and loop at the left is called as D arm, D psi arm. There is a T arm. There is an anticodon site. Anticodon sites are mostly at the 35, 36, 34, 35 and 36 bases. Okay, so in and around 100 bases these are there, 70 to 80 bases along this tRNA, small molecules which are internally folded, a single stranded RNA internally folded to give life to this beautiful stable structure. These are typically around 70 to 80 nucleotides in length, and one loop which is called as the anticodon loop here is at the 34, 35 and 36 bases. The upper stem which is there, this upper stem is known as the acceptor stem because here it accepts the amino acids. And like this, some 30 to 40 tRNAs are there, 64 codons, 30 to 40 tRNAs, and each tRNA with amino acid, it's called as a cognate tRNA or a charged tRNA. And one very interesting factor of this tRNAs in making it stable, this is the same structure here at the end, but this is a ribbon model. This is a ribbon model. This is a simple diagram, and this is the diagram showing the bases, specific bases which are present. There is, now you see this colored molecules. There is psi, this is psi which is pseudouridine. There is T which is ribothymidine. There is D which is dihydrouridine, and there is somewhere I inosine. Now you see I is very interestingly present in the 34, 35 and 36 group, and inosine is responsible for Wobble hypothesis. If you recall, I had given you the inosine molecule. From where does this inosine molecule come? It is a stable structure present in the tRNA, and this has been modified during the course of evolution. Okay, there are methyl groups present on certain very specific bases, methyl 1, 2, so on and so forth. So this all forms the cloverleaf model, and these bases, what makes this stable structure is the bases. There are certain bases which have been modified, and they occur at several randomly distributed positions throughout the tRNA. Having said that, it may look random, but evolution has chosen them to be a very specific point, and these modified bases make the tRNA unique and stable. These are not present in the Watson and Crick pairing, but these are present in the nucleotide pool, and they are chosen to form this cloverleaf model. So this L-shaped tertiary structure, the ribbon model of the bioinformatics structure or the computer model, you can say they all tRNAs, they have a cloverleaf secondary structure, and they are folded into a L-typed secondary tertiary structure. So from this 5 end to 3 end, if you just open it, it will form a long chain of mRNA, but you will just release it, internal folding, they will fold into a L-like structure. 4 double helical stems are there, 1, 2, 3 and 4. 4 double helical stems occur. 3 of these loops have 7 to 8 residues at their end. The amino acid is attached to the ester linkage to the 2' hydroxyl or 3' hydroxyl group of the A residue, that is the acceptor stem. Many residues are modified in tRNA, and some modifications I have shown already in the figure in the last slide here. Here you can see the modified bases. So you can pause this and see anytime. So now once you understood the structure of the tRNA, what is the function of the tRNA? It's very clear that the function of the tRNA is to bring a amino acid at the site of protein synthesis. This will continuously go on bringing. One will bring, another will bring, both will form a polypeptide chain. It will remove, third will come, this will be removed, fourth will come, this will be removed. What happens with this removed? This goes to the amino acid pool, gets charged, brings another amino acid. So it is a cycle process. The charging of the tRNA is very important here. This takes place somewhere else from the site of protein synthesis. This charging does not take place on the ribosome or afterwards. It is charged, already charged in the cytoplasm, where maybe in the amino acid pool. There the tRNA will go, it will bring small corresponding amino acid corresponding to its anticodon. Anticodon and the amino acid, they are matching the codon. So a correct amino acid is brought. This is always almost a highly accurate process, high fidelity process, and this minimizes the errors of mutation. So see what happens here, charging of a tRNA. Now this is a tRNA, which I just L-shaped tRNA molecule, and this is the enzyme. This enzyme is called as amino acid tRNA synthetase. Amino acids are linked in ester. Read the slide, you can pause and read. I am explaining how the charging takes place. There is a amino acid tRNA synthesis synthetase enzyme mediated synthesis. See, listen again. Amino acid tRNA synthetase mediated synthesis of charging of a tRNA takes place. What is charging? Adding the correct amino acid to the correct tRNA. A tRNA without amino acid is called as an uncharged tRNA. A tRNA with a wrong amino acid will be called as a mischarged tRNA. So the first step in protein synthesis is somewhere in the cytoplasm, charging of the tRNA. Amino acids are attached in ester linkages to the 3' terminus of the tRNA, forming amino acid tRNA, and this is mediated by amino acid tRNA synthetases. Amino acid tRNA synthetases are highly accurate. They work with high fidelity. No errors are there, and this helps in minimizing the translational errors. This is the first step. Now you see the active site. The active site here, see the laser pointer. The active site on the amino acid tRNA synthetase corresponds to the substrate molecule, that is tRNA. So even the anticodon site is fitting in the enzyme, and the anticodon and the codon binding takes place afterwards, but here the accepting of the tRNA by this amino acid containing amino acid tRNA synthetase is a very important energy requiring step, and this is continuously going out in the cell. In the step 2 of this charging of the tRNA, the amino acid is added to the growing protein chain based upon the codon and the anticodon, that is tRNA's anticodon and the codon of the mRNAs. Bacteria have 30 to 40 types of such tRNAs, whereas in eukaryotes 50 to 100 molecules may be present. A given amino acid often can be carried by more than one species of tRNA, and that is what is degeneracy of the code is all about. Okay, so the enzyme which carries out this ATP driven reaction, which is known as amino acid tRNA synthetase. See the final mechanism now. This is the enzyme containing sites. This is the tRNA site, this is the amino acid site. So this is right now, this is a uncharged tRNA. See here, this is a uncharged tRNA. Now if you just see here, this is a uncharged tRNA, and this is, sorry, that is a uncharged tRNA synthetase enzyme, means which is not containing a enzyme. This is mediating the charging of the amino acid with tRNA. So this is the enzyme and this is the tRNA. So this will get added here, and the final, before the final product is formed, this is the E plus S reaction. And you see here, this is the P. This is the P and this is E. E is released from the P and this is the E plus S. This is substrate. This is a substrate. This is enzyme. This is substrate. This is enzyme. And this is the enzyme. So if you just see here now, the enzyme contains a site for addition of tRNA. It contains site for addition of amino acid. In this example, tryptophan is given, which contains a amino group and a carboxylic group. So addition of this amino acid to the amino acid tRNA synthetase is a ATP driven reaction. Okay, it binds. Now you have an enzyme which is containing a amino acid. It is ready to act with its substrate, the tRNA, anticodon specific to that tryptophan. This tryptophan recognizes the tRNA. Anticodon specific to tRNA will be bought. There will be a E plus S reaction, and now here what happens, the tryptophan is transported on to the tRNA. So here tRNA now contains this amino acid, and charged tRNA now will travel to the site of protein synthesis, that is the ribosome. Got it? So this is all what it happens with the charging of the tRNA, and as it gets charged, as there is charging of the tRNA, it should be remembered, like these reactions, there are around 30 to 40 types of reaction taking place, and these 30 to 40 types of reactions are not taking place at the point of protein synthesis, but they are taking place somewhere in the cytoplasm, and all this helps in a smooth translation process to occur. So you summarize all these three reactions in one. Charged tRNA is being formed here. In the first step of the reaction, the amino acid is activated. The amino acid reacts with the ATP molecule to produce an intermediary carboxyl end of amino acid getting attached to the AMP. Two terminal phosphates, that is the pyrophosphates, are cleaved from ATP in this reaction. In the second step, you can see the amino acid AMP complex remains bound to the enzyme. It remains bound to the enzyme. tRNA next binds here. The tRNA next binds to the enzyme, and in the third and the final reaction, the transfer of amino acid from AMP to the tRNA takes place, producing a charged tRNA and a AMP. The charged tRNA, this charged tRNA now consists of a very specific amino acid attached to the 3' acceptor stem of the RNA, and it is ready to travel to the ribosome for protein synthesis. If this charging is perfect, it is called as a charged tRNA. If this charging is wrong, there is a wrong incorporation of amino acid, it is called as a mischarged tRNA. Rarely that happens. And if there is no charging taking place, or a tRNA without a amino acid, is called as uncharged tRNA. So these are the various structures which you may find in the books. These are just depictions, mere depictions. So this is icon. What we just saw is an iconic structure of tRNA. This is the 3D space filled model created by the bioinformatics technology. This is the ribbon like model, and all these are generated from the structures observed under electron microscopes for real tRNA molecules. And this is the cloverleaf model, a 2D simple diagrammatic cloverleaf model. You may be encountering any of these models in your books, so I think it should be easy on your mind to understand whatever the structure is there. In one of the animations, they show this 3D space filled model. I have given the link below of Vehi Educations, DNALC, you can see that beautiful animation is given, and they employ this 3D space field model to show the translation. Some YouTube links also I have given in the below. You can watch them for a clear view of protein synthesis. Now here the codon and the anticodon binding of tRNA with mRNA is very interesting. You should know this. The first base on the tRNA or the third base on the mRNA, they have always wobbling situation, wobble pairing. It's just like a plug and a pin in the plug pin. The third pin, which is of the earthing, is important but not that significant. Even if the third pin is not there, these two, neutral and phase, they can carry out the whole electricity process, but this is needed for stability. Just like that, one base is wobble, it is not that important. So the codon anticodon base pairing, codon and the anticodon base pairing is to be understood here. The hydrogen bonding between, here you see the laser pointer, the hydrogen bonding between the first and the second positions of the codon, first and the second position of the codon, and the third and the second position of the anticodon, third and the second, first and the second on the codon, third and the second on the anticodon, nearly always occur via Watson and Crick pairing. So 1 and 2 on the mRNA and 2 and 3 on the tRNA are always Watson and Crick. It is always with either 3 on mRNA and 1 on tRNA, or 3 and 1, 3 on mRNA and 1 on tRNA. They have a wobbling situation. Base pairing between the third position of the codon, focus on the laser pointer, and the first position of the anticodon are less constrained. They do not follow Watson and Crick pairing, they have Wobble pairing. Wobble base pairing has a significance. It reduces the number of tRNA genes that an organism must take to carry out translation. If this also followed Watson and Crick pairing, then there would have been need for 64 tRNAs. In evolution, they would have made 64 tRNAs. But somehow evolution finds a way for efficiency. Less the best. Best processes in less resources. That is how the survival of the fittest mechanism comes into theory, you know. As many as less resources and maximum capacity of functioning. So Wobble base pairing, it reduces the number of tRNA genes that an organism must make to carry out translation. It also helps in protecting against mutations that might inactivate the tRNAs. So you see the table here now, we will see two situations. This is the first situation, 1, 2, 3 on the mRNA. So 3 on the mRNA and 1 on the tRNA. So in the first table, I am talking about the first anticodon on the tRNA, which may wobble. See, there are two, as we are talking about 3. So if I am talking about this wobbling, either this may wobble or this may wobble. Okay, so this is a mRNA, this is a tRNA. So tRNA, I have 1, 2 and 3. Okay, this is 1, 2, 3 of the tRNA, and this is 1, this is 1, this is 2, and this is 3. So 1 and 3, this is 1 and 3. So 1 may be a wobble or 3 may be a wobble. That is what has to be understood here. Try to understand. So if the bases are in the first or wobble position of the anticodon, if these bases are in the first wobble position of the anticodon, what can be the probabilities? C, A, G, U, I will be there. C, violet to violet color. C, cytosine, adenine, guanine, uracil or inosine molecule may be there. Then the tRNA may recognize codons in mRNA having these bases in the third. C can recognize G, normal, okay. A with U, G with C and U both. U in the tRNA with A and G, and inosine molecule can recognize C, A, U. Now on mRNA there is no inosine. Third on the mRNA, let's see. What are the possible bases on the mRNA? C, A, G and U only. Whereas on the tRNA, C, A, G, U and inosine molecule, inosine has the highest wobble. It can recognize cytosine, adenine, uracil, all. Okay. So this is how a tRNA functions comes, recognizes. Now the mode of action of tRNA at the molecular level I have explained here. You should understand this when we will see the whole process in detail. Okay, so tRNAs and ribosomes, they are made for each other. They have a significant attraction, they have significant, what you can say, space for each other, and they just accommodate, remove, it goes comes as its own house. It comes in the den, leaves amino acid, goes away, comes again. When the process is at great speed, 30 to 50 molecules are added per second. These tRNAs, the site for the tRNAs is the 50S molecule of the ribosome. So large subunit, small subunit. Large subunit accommodates the tRNAs. So if you take a sectional plane of this large subunit bound to small subunit with the mRNA, that is a translation process in action, cut it, you will find like this. You will find a structure like this. That is a sectioned ribosome which contains E site, empty site, P site, peptidyl site, A site, the amino acid site, and the mRNA. There is a space for mRNA. mRNA is bound to the 30S subunit, and the tRNAs will be bound on the 50S subunit. Ribosome has multiple tRNA binding sites. P site binds to the tRNA attached to the growing peptide chain. A site, it binds the tRNA, it binds the tRNA carrying the next amino acid, and E site is an empty site. You see, if you just imagine, there are 3 seats. There are 3 seats, but the situation is that once one person sits on this seat, another person comes and tells him to move to the next seat. So he comes onto the middle seat. Here he comes. Now the third person comes and he just pushes the third onto the third. Now there are 3. The fourth one comes. As the fourth one comes, the last person is removed off. So this process, imagine in speed. Speedily this thing happens, and as this thing is happening, you are having a last site which is almost always empty, because that is going away. Coming, last is empty. Coming, pushing, last. That is called as the E site here. It follows a particular direction. We will see that. So ribosomes has 2 primary functions: to decode the mRNA and form peptide bonds. So you see, this is the ribosome, 30S, 50S, and the mRNA. And I am just talking about the function of tRNA here. We will see this elongation process in great detail in the next part, that is part 2 of this process, called as the prokaryotic translation. So here you just focus on tRNA. What is tRNA doing? So there are 2 sites. The first site, it has what amino acid. So as it moves ahead, as this moves ahead, automatically the first site tRNA comes to the second site in the middle, and A site is free. In the A site, as you can see, another tRNA with correct amino acid, chart tRNA comes. Again, as it moves forward, the second amino acid comes to the middle and the first amino acid goes to the E site. A third amino acid coming, displacing. This whole process is called as translocation, and during this, what happens when the last E site tRNA has to go away, it gives its amino acid to the incoming tRNA, and the need for the peptide bond there is felicitated by the 50S subunit. That is, ribosome felicitates that. So this is how charged tRNAs, they accumulate these amino acid chain. With each step of the tRNA getting displaced, there is one molecule of amino acid bound on the top. So 1, 2, 3, 4, 5 tRNAs continuously going and coming and going, coming and going out, and each step giving one amino acid gives rise to a polypeptide chain. So this transfer of the amino acid from one tRNA to another tRNA. See another enzyme now, very interesting enzyme, peptidyl transferase. This is again a function of tRNA. We are seeing tRNAs. Peptidyl transferase is the enzymatic component of the ribosome which forms peptide bonds between 2 amino acids. So it not only transfers, but it forms a bond. Peptidyl transferase, transfer and formation of peptide bond, a very interesting reaction which gives stability to the chain of amino acids. Okay, this is how the tRNA they function. The translation in prokaryote requires initiation of the translation and an initiation complex is formed. It requires an initiator tRNA. What is the initiator tRNA? Initiator tRNA is the tRNA which brings N-formyl methionine. What is N-formyl methionine? This is the amino acid recognized by AUG codon. In genetic code characters, we have seen that AUG is the initiating codon. Always, almost, universally, 100% of the times, AUG when it comes at the first, N-formyl methionine will be added. If it comes in the middle it is not initiating codon. There methionine will be added. Remember this point. When AUG is the initiating codon, N-formyl methionine is added on the tRNA or a tRNA. There are two types of tRNA for AUG codons. Anticodon containing tRNAs will be two types for AUG. One is N-formyl methionine containing tRNA, which will always come when AUG is the initiating codon. And if AUG is present somewhere in the middle of the polypeptide chain, not a initiating codon, then a tRNA with methionine will come. Okay, so this is the initiator complex. The initiator complex is of 50S, 30S, mRNA, tRNA with a N-formyl methionine and a AUG codon and the mRNA. So this is an initiation complex, and this initiation complex involves initiator tRNA charged with N-formyl methionine, small ribosomal subunits and the mRNA strand. The ribosome binding sequence of mRNA is complementary to that part of the tRNA. Therefore, it comes and attaches. These all are temporary Watson and Crick pairing. Only the permanent type of Watson and Crick pairing is present when DNA forms a DNA and a double-stranded DNA is converted into quadruplet strand of DNA. There the hydrogen bondings are permanent. Here you see the mRNA has some bases on which a 30S subunit comes, that has some complementary sequence. That is a temporary one. Once the process is over, they dissociate. All these associations are brought together by some proteins. I will explain those things also in the end. So finally, coming to the function of tRNAs in this lecture, I would like to tell you there are 5 major stages of protein synthesis in E. coli. All these stages we will be seeing in our next discussion, part 2 of this lecture. But I will just give you a brief summary of how these 5 stages are worked out. These 5 stages are: the first is activation of amino acids. tRNA activating enzyme activates the amino acid producing a charged tRNA, just now as I have mentioned this process. The first stage is activation of amino acids. 20 amino acids are there. 20 amino acid tRNA synthetases will be there. 20 or more tRNAs will be there. Each step requires ATP and magnesium ions. Second stage of translation or protein synthesis is initiation. This initiation requires, now what I am talking about here about the translation can be explained for short notes. For 4 marks this is sufficient what I am saying, but for more understanding you will have to listen to my next lecture. So how does protein synthesis takes place? It takes place with charging of the tRNA, activation of amino acids. Second stage is initiation. In this initiation, as you can see, this mRNA chain is needed. N-formyl methionine tRNA, see the cursor pointer. Second requirement is a tRNA, charged tRNA with N-formyl methionine is needed. Initiation codon AUG on the mRNA is needed, and subsequently the anticodon UAC containing N-formyl methionine will be needed. A 30S subunit of ribosome is needed, 50S subunit of a prokaryotic ribosome is needed. Now there are certain proteins called as initiation factors. Initiation factor 1, initiation factor 2, these are needed. The main energy currency here is guanosine triphosphate, GTP. GTP molecules and magnesium ions are needed, and once all these assemble and once the ribosome starts to move, that is called as translocation. Once the ribosome starts to move, it produces polypeptide chain. The third stage is elongation. You see only this much. The elongation. In elongation, the tRNA displacement, which I just said, it takes place. Each charged tRNA brings an amino acid and a polypeptide chain is synthesized. Elongation of amino acid chain, polypeptide. The elongation refers to formation of a polypeptide chain. In transcription, elongation refer to elongation of mRNA chain. In translation, elongation refers to elongation or lengthening of polypeptide chain. So what are the requirements? Here functional 70S ribosome is needed, initiation complex is needed, amino acid tRNA specified by codons are needed, and as initiation factors are needed in initiation, elongation factors are needed in elongation. These are elongation factor TU, elongation factor TS, elongation factor G. You can call them as elongation factor 1, elongation factor 2, elongation factor 3. Termination, you see here the fourth stage is termination. Termination takes place with you can see nonsense codon. Okay, termination it UAG, as you can see here the termination is UAG. What happens with UAG is that this UAG codon on the mRNA does not have a tRNA. Why it is called as a nonsense codon? Because it doesn't make any sense, it cannot be read. Or after adding all this polypeptide chain, as I said, you can explain these in short notes of protein synthesis, but here my main discussion was to make you aware of tRNAs, and I am just summarizing the protein synthesis in short, which we will see in detail in our next lecture. So the fourth stage is termination and release of the polypeptide chain. Termination codon is there UAG, which does not have any tRNA, so it recognizes release factors. So release termination will require release factors RF1, RF2, RF3. So termination takes place, and after that, once this polypeptide is terminated, see this figure now, these three figures, the folding and post-translational processing of this polypeptide chain takes place. Unwanted ends are trimmed off. Depending upon hydrogen bond, disulfide linkage, hydrophobic, hydrophilic linkages, and all the bonds and linkages which you studied in chemistry and whatever is possible here, it has an internal folding. Specific enzymes, cofactors and other components are needed, and some parts are removed, some parts are added. Proteolytic processing takes place, modification of terminal residues takes place, and attachment of phosphate group, addition of methyl group, addition of carboxylic group, carbohydrate or lipids or prosthetic groups may be added. Means you have carbohydrate based proteins, you have lipoproteins, so on and so forth. All this modification is the final stage in the cytoplasm, takes place somewhere else, and you have a fully functional protein. If it has active site, this protein may function as an enzyme, and this is how the protein synthesis takes place. So if you have got the idea of how this protein synthesis is taking place, then let us now just see the small process in animation. See here, so you can just see, I'll try to put it once again. So this is mRNA chain, 50S, 30S ribosome binding, tRNA is coming, bringing amino acids, translocation taking place, and a protein formed. See it once again, 50S, 30S, ribosomes accommodating tRNAs, charged tRNAs, then there is a peptidyl transferase enzyme finding peptide bonds, and a polypeptide is released. Okay, so this is how the whole protein synthesis takes place, which we will see in great detail in our next lecture. Right now I hope you understood the role of tRNAs. It's very important. So you have seen structure of RNA polymerase, structure of ribosomes, structure of tRNAs. These are the important molecules required for protein synthesis, and mRNA, tRNA, rRNA, these all assemble to give rise to a functional polypeptide chain. So the next part of my discussion will be prokaryotic translation. Hope you go through this lecture quite a few times and make this concept very clear, so that you will understand the next most enjoying part of life, that is translation. Translation is life, let me tell you. Even after a person is dead, they say that some reactions in his body are going on. Means once he is brain dead, he is dead, still for some few hours his body processes are on. Such is the power of this metabolism and translation. So how does this translation takes place? I will be explaining this in my next lecture. Till then, happy studying, happy enjoying genetics, and hope you have newer insights, newer knowledge and newer information in genetics with me. Thank you. See you once again.